My name is Scott Hamilton-Brehm. I'm an assistant professor of microbiology at SIU. And I got my degree in biochemistry, and I focus on the biochemistry of microbes, specifically environmental microbiology. So the microbes that live in the environment from air, water, earth, and specifically I work with microbes deep underground. When I was very young, I had a fascination for the extremes, the organisms that lived in boiling water or deep out in space, if that's possible. And so that kind of drove me to learning biochemistry of microbes, actually, because at this level they're a little more simplified, so it allows you to kind of tease apart to try to understand the secrets of how does a living thing live in, say, boiling water or deep underground or live by radiation. And so there was actually very few places as far as universities to go to get a degree, but I went to University of Georgia, where I worked with an organism called Pyrococcus furiosus, which is, literally means the burning fireball or the thrashing fireball, and it grows in boiling water. And so that's where I worked, did my graduate work, and graduated from there, and then from there did postdocs at Oak Ridge National Labs and the Desert Research Institute in Tennessee and Nevada, still continuing working with extremophiles. And then I brought all that experience and that access here to SIU, where I continued working with some subsurface microbes, these extremophiles. And not only do I work on, all my students in my lab, undergrads, graduate students, work on various points of understanding how life survives in the extremes. Right now, within the scientific community, we know very little. There's very little knowledge. So this type of research is accessible to undergraduates, graduate students, basically anyone who's willing to come into the lab, get their hands dirty, make up media, and start counting, watching the cells as they grow and characterizing what they can and can't do. That's what my lab does. A typical day for an undergrad would be to grow microbes in my lab and then to transfer them. So in order to do that, I'm gonna have to sterilize. That's ethanol. The flame. This is a sterile syringe with needle. I'm going to take a sample from the bottle. That's the first bottle, and that's the second bottle. So this organism has been successfully transferred to 2 new homes. Now we take the microbes' new homes here and put them into the oven. Once in the oven, they'll warm up, and this will become the optimal temperature at which these microbes will grow. And an undergraduate would be monitoring the progress of how they grow. If we discover something new, we would also start extracting DNA and doing PCR and other means of taxonomically identifying what these microbes are. Other students would be interrogating microbes based on their metabolic products. What are they consuming? What are they eating? And by doing that, you can use instruments like an HPLC, high pressure liquid chromatography. Each one of these samples are coming from different microbes. And so with the instrument like this is a high-end industrial piece of equipment that looks really good on your resume, but it allows us to be able to interrogate microbes and find out what are they producing when we're feeding them different substrates. Sometimes we go out in the field to collect our samples for the microbes. And in order to go out in the field, we have to bring equipment with us. And it could be something very complex like a pump, or it could be very simple, just like markers, gloves, and sterile water. But then also we have to then collect the samples and put them into sterile containers and then lug them back into the lab. And this is a very complex process in the sense of just taking this equipment out there and then bringing it back, labeling, and then processing, and then trying to figure out what we found, what we got out in the field. All right, here we're at our site, and we are now drilling into the ground and removing sections of ground, earth. And inside that earth column are microbes, and we're going to take them and we're putting them into these containers. And as we get them to the containers, we're going to freeze them and bring them back to the lab. Locked away in this sediment column should be microbes in there that will give us an answer to one of the questions we have, which is, where's all the nitrogen going? This area is starting to lose tons of nitrogen, and we're not entirely sure why. If this is interesting to you, please contact me, and there are openings within the lab, and more information, click on the link below.